My name is Ehrin Armstrong. I'm the medical director of the Adventist Heart and Vascular Institute in St. Helena, California, and the national PI for the upcoming SELUTION4BTK IDE trial. As others have discussed, the solution SLR Balloon has a number of unique characteristics that I think are going to make it an important advance in the treatment of tibial disease using lima solution. This includes a proprietary microreservoir technology that allows microreservoirs to combine sirolimus and a biodegradable polymer, thereby allowing a sustained release of sirolimus over the course of 90 days. These microreservoirs are essentially a miniaturized drug delivery that allows a pharmacokinetic release profile comparable to best-in-class drug-eluting stent and a sustained therapeutic effect for up to 90 days. And finally, the balloon includes a cell adherent technology that enhances drug retention and bioavailability, allowing for lower drug dose concentration on the balloon surface at 1 microgram per millimeter squared. This helps overall optimize the transfer of the drug to the tissue and the cellular uptake of sirolimus. This video here just demonstrates what that looks like. You can essentially think of the balloon as having thousands of these little micro reservoirs that are little drug delivery units to deliver sirolimus to the tissue when the balloon is inflated over a period of a few minutes. So the SELUTION4BTK IDE trial, as a general overview, is a multi-center randomized trial with the objective to demonstrate superior efficacy and equivalent safety of the solution SLR 0.014-inch drug-eluting balloon compared to uncoated balloon angioplasty for the treatment of tibial disease in patients with critical limb ischemia. As I mentioned, it's a prospective, multi-center, single-blinded trial. It's going to involve 40 sites in the USA, Europe, Singapore, and Japan, and enroll a total of 377 patients, with 50% of those in the United States. Importantly, follow-up will be up to 5 years. The primary endpoint is a composite freedom from target lesion occlusion, above the ankle amputation, and clinically driven target lesion reintervention at 6 months. The safety endpoint is the freedom from major adverse limb events and all-cause perioperative death at 30 days. And as I mentioned, I will be the US PI, Marianne Brodman is the EU PI, and Professor Yip is the PI for Asia. So here's an overview of the trial design. Of course, because we are dealing with tibial disease, we have to take into account the fact that most patients with critical limb ischemia will have some type of inflow disease in the iliac or SFA segment. So the inflow disease, of course, will be treated first, and the successful inflow treatment can include the use of any non-drug-coated balloon or drug-eluting therapy, including the use of bare metal stents or atherectomy for inflow treatment. Drug-coated balloon use is allowed in the SFA segment, but it has to be with a solution balloon size to the SFA so that any drug that is used is sirolimus and not paclitaxel. Once the inflow vessel is treated, the below-knee target lesion can be treated with vessel preparation. And then if there's successful preparation, the randomization occurs 1-to-1 to the solution SLR balloon versus balloon angioplasty, so that there is a direct comparison from balloon angioplasty to the use of the solution balloon. Here are the sites outside of the US. This includes current site list in Hong Kong, Singapore, the United Kingdom, Netherlands, Germany, France, Switzerland, Austria, and Italy. In the United States, we have a number of sites that are becoming initiated as well. This includes a number of sites on the East Coast as well as some in the West Coast and the South. So I think we have a really robust set of centers who have a lot of experience with enrolling critical limb ischemia trials and are able to follow up these patients well for the full 5-year follow-up, which will be critical to demonstrate the long-term efficacy and safety of the balloon. So thank you very much for your time, and look forward to moving forward with this very exciting trial. Thank you.
Thank you very much, Ehrin, for the summary here of the IDE trial. And I think we need to discuss 2 points here on this IDE trial. First of all, atherectomy is allowed. I think that is absolutely interesting here. What is your standpoint about having an atherectomy in an IDE trial? That is interesting.
Well, it'd be one of the first, wouldn't it? So,
yeah.
I mean, I think for tibials, I think it's actually quite a good thing. But I think the effect will be seen more like between 6 and 9 months after DCB than actually right within the first 6 months. But very interesting. I think you'll get more lesions to be crossed and more enrollment as well. So I think it's an interesting concept, and I'm looking forward to that subgroup analysis.
But please, no peripheral embolization during atherectomy, yeah?
Slow.
Exactly. You're at risk.
Yeah, this could be the risk. And the second point, interesting, is that we are allowed to use sirolimus solution technology as inflow treatment. This is also unique here, and this will also be interesting in the dosage measurement, which we are required to do at the end, so that we know about the dosage of the patient, including inflow treatment with the solution technology. Yeah.